Great, so hopefully everyone can see and hear me A-OK. My internet is a little bit laggy, so that if it gets significantly behind, if someone could just let me know, that would be great, and I can pause so it can catch up. So I'm going to be talking today about some work we've been doing in our lab, looking at how perception of colour discrimination in particular lines up with commonalities that you find in natural scenes. And this is some work we've been doing with some collaborators in Ecuador and Asper, who's just moving to Oxford now. So we're particularly interested in how the chromatic environment you live in currently might impact your perception of colour and the timescale that this might happen over. So if, for example, you grow up in a rainforest environment like this one and you live in that environment, does your colour perception align in a way that is quantitatively different with your environment, in a way that is different to people who might grow up in either of these two cities, for example? And we do know that there is some evidence of alignment between natural scenes and colour perception. And one of the ways we know this is thanks to this commonality that's regularly found in natural scenes, which is this elongation along the blue-yellow direction from counterpoint in colour space. So if you take an image like this one and you plot the chromaticity of every single pixel of this image in this space, you find that it tends to show a greater amount of variation along this negative diagonal, which corresponds to bluish to yellowish, than it does in the opposite direction, so reddish to greenish. And this is common across lots of different types of scenes and across lots of different image sets that exist there, out there, including hyperspectral image data sets. And it appears that adult saturation thresholds align with this commonality that you find in natural scenes. So if, for example, you asked a participant to do a target detection task where they have to spot a chromatic target in amongst a field of grey distractors, where this chromatic target varies in saturation along each of these 8 spokes, what you'll find is that participants need blues and yellow hue to be more intensely coloured, so more saturated, in order to reliably detect it. And we can plot that by measuring saturation thresholds for each of these 8 axes and then putting it in a space like this one, where distance away from the centre shows you the size of the threshold. So further away means that that hue needs to be more saturated for your participant to reliably detect it. And interestingly, so this is an adult perception, but we also find it in infant perception. So here I am dutifully eye tracking a baby who's doing a target detection task, where a coloured chromatic target appears a certain distance away from wherever the infant is fixating. And if the infant can see the target, it will make an eye movement towards it and it will turn into a smiley face for them. Whereas if they don't see it, the next child's presented and it carries on. So the infant sees a range of different hues at different saturation levels. And just like with adults, we can plot psychometric functions in order to measure their discrimination thresholds. So we can read off the point where our infant participants who are 4 months old can reliably detect that particular hue at that particular saturation. And what we find, like with adults, is that there's this elongation along the negative diagonal in this chromaticity space. So blues and yellows need to be more saturated in order for our infant participants to be able to reliably detect them. And this suggests to us either that this tuning into the environment happens incredibly quickly, so in the first few months of life, or that it's evolutionary bias that exists in our visual systems. And it kind of begs the question of, well, is this set for life, or do you tune into your unique chromatic statistics that you come across in your environment? So why would it be the case that greater variation along this blue-yellow diagonal would result in poorer discrimination in both infants and adults? It's not something that can be explained by cone-opponent mechanisms alone, so it's not just due to poorer discrimination along one particular axis. If that was the case, then you would expect to see that red and green thresholds were also higher, because they are also making use of the same mechanisms that underlie colour vision as the blue-yellow direction. So an alternative explanation is related to efficient coding, and in particular a range accuracy trade-off. So the mechanisms that we use to represent a particular dimension, in this case saturation, has the same range that it can cover but the variation, what's out there in the world that it actually has to represent might be very different. So in this example that we're talking about here, the stimulus dimension that we want to represent is this large variation in blue and yellow versus a much narrower variation in red and green in the opposite direction. But both of the mechanisms underlying these processes have got the same range to do it with. So that means that you have to take larger steps within this negative diagonal in order to be able to more accurately represent it to efficiently code it. So efficient processing is all about representing the environment as it really is, as accurately as possible, given the constraints that the neural system is currently under. And I'm going to describe this idea in more detail in relation to one particular chromatic scene statistic, and that's the way that saturation accumulates across different hues in our environment. So to do that, if we imagine that we live in a very, very simple pixel world where we just have two hues that only vary in saturation, so it might look something a little bit like this, and we've got a very limited number of neural mechanisms to encode our environment in. We can plot the way that saturation varies in our environment using a figure like this one. This shows you the cumulative frequency of saturation. So it varies from 0 to 1, where 1 would mean that every single pixel's saturation has been accounted for, and 0.5 would mean you've accounted for 50% of the pixel's saturation levels. So this isn't about the amount of pixels of a particular saturation or of a particular hue, but it's about the variation that you find in the pixels of a particular hue and saturation. So it wouldn't be very efficient for our neurons to code these two distributions in exactly the same way. That wouldn't be an accurate reflection of what's happening here. So we would expect with efficient coding that the neural response would match the shape of our cumulative frequency distributions. So we can use this y-axis as a proxy for neural response. So when we're looking at these two distributions, we can see that they're not the same. The red line is much steeper than the blue line. So the range of saturations that you're likely to come across for your reddish pixel is much narrower. It accounts for all of its saturation, all of the pixel saturation levels at this low saturation point. Whereas the blue line takes much longer to account for every pixel that's in our world. It's got a more even spread across the possible saturation levels that are in the environment. So how does our visual system, how would we expect our visual system to represent this? Well, if we take a single level of neural response, so this is 0.4 on this axis or 40%, we can, just like we would with a psychometric threshold, read off how saturated the world is at that point once we've accounted for a particular proportion of pixels or a particular level of neural response. And to make it even simpler, let's say that this 0.4% point on this y-axis represents 4 steps or 4 levels in neural response. So for our red hue, we have 4 levels that we can use to divide up this saturation range. Whereas for our blue hue, we still have the same 4 levels, but we've got a much wider range of saturation that we need to cover. So the steps that we take are much larger, and that's why discrimination thresholds are poorer. Whereas for our red hue, because the steps we take are much more fine-grained, it means that our discrimination for this hue would be better, we'd expect. So how does this relate to the variation that we find in natural scenes? Well, in this example, we would expect that the elongated blue-yellow matches best the distribution kind of mock described here by this blue function. There's a greater range of variation that needs to be accounted for with this limited neural mechanism. Whereas in the opposite direction, there's a much narrower range of saturation levels that need to be accounted for, but using the same number of levels of the same neural response as we use for the blue. So how would we test this idea that we are efficiently coding our environment so that our perception lines up in an efficient coding way with what is out there in the world? Well, the way that we've done it is we've compared the chromatic statistics of 3 different environments to the perception of people who are living in those 3 different environments. And the perception in this case is by measuring saturation thresholds. So the three different environments we have, so one is a rainforest. So this is a rainforest, which is in the very north of Ecuador in the Esmeraldas. And then we have two cities, so Quito, the capital city of Ecuador, and Brighton, which is a city on the south coast of the UK. And qualitatively, when you're in the Esmeraldas, the environment feels very different to being in a city. It's noticeably lush and green, and there's not much grey tarmac and things like that. The two populations who are taking part in this study who live in Ecuador are part of the same indigenous populations, members of the Chachi community. So they have some more recent shared genetic history than people living in Brighton in the UK. So the first step in this is to quantify the environment that these participants are living in. So our participants wore head-mounted GoPros that were calibrated. So you can maybe just about make out on this person here that they've got a head strap on and their GoPro is attached to their forehead. And the camera is set up to take a photo every 30 seconds for 3 hours, because that's the duration of the battery life, and participants are told to go about their day. So here you can see a quick representation of what these images look like. And so from this, we get thousands of images. For the image analysis I'm going to be talking about today, we've got a random sample of about 350 images from each location. So here's a snapshot of what the image sets look like. So here's an example of some of the images from each location. Something to remember is that obviously working with the GoPro camera, we're constrained slightly by the RGB gamut, but we're hoping in the future to follow up on the statistics that we find using hyperspectral imaging, either in the same environments or in very similar environments, paired with the GoPro as well. So now we have all of our images. How do we analyze them? So for each image that's in the set, we plot the chromatic distribution of pixels which are found in that image. So here is the distribution of this image, for example. You might just be able to make out a tail there of the green foliage that's outside the window. And once we've done this, we can then work out which of these 8 hue bins the pixels fall into. So the hue bins that we're using are defined by these black outlines that you can see here, and they're centered on the axes that we then later measure discrimination thresholds along. So now we have the number of pixels which fall into each hue bin, and we can estimate the saturation of them. So white points are very important when you're determining saturation, because in Cavoyton chromaticity diagram, saturation is defined by distance from white point. So for the data I'm presenting today, we used equal energy white as our white point. We did also collect data going through these images and the whitest pixel, so that each image would have its own unique white point. But we found that when you do that, it doesn't have a big impact or really any impact on the cumulative saturation distribution that I'm going to be talking about from now on. So for simplicity, we use the same white point for every image across all 3 sets. So now that we've got both the number of pixels and how saturation varies, we can plot our cumulative saturation distribution, which looks something like this. And from this, we'd be able to read off how saturated, the maximum saturation that is accounted for by, say, 50% of the neural response. So these images are a little bit hard to intuit, so I'm just going to take you through a couple of examples now. So here's an image where you've got a big range of a particular hue that needs to be accounted for, and you have some very highly saturated examples of that hue. So this red line here corresponds to the reddish hue bin. And so you can see that it takes quite a while to reach, that it takes quite a while to reach maximum saturation, the point where it has accounted for the saturation of all of the pixels which are in that hue bin. Whereas in comparison, the other hues are much, much steeper. There's less variation in this image of those hue bins. And here's another example where the hues are pretty similar in their response, perhaps with the exception of yellowish, partly driven by this intensely yellow door over there, and also the colour of the ground. And just like we would with a psychometric function, we can read off a particular point on this y-axis to get a measure of what the maximum saturation level of this real world is out there. And we can plot that point in a space similarly to how we would our psychometric discrimination functions. So for example, you can see that this yellowish point is further out. It has to account for a greater amount of variation in saturation. So the point is much further out. Whereas in comparison, this pinkish hue only has to account for a much narrower band of saturation with the same level of neural mechanism. So it's much closer to the center. So then we'd be able to compare this distribution to our behavioral data that we collect from the participants who live in these three environments. So just as a reminder about what our task is, participants do this 4AFC task where they have to pick a colored target out from a field of gray distractors, and the chromatic target varies along each of these 8 white axes that fall in the center of the hue bins that we're using for our image analysis. So we can find the point from plotting psychometric functions where participants can reliably detect the target, say 50% of the time, and read off their threshold. And then again, the further away a point is from the center of this ellipse, the more saturated that color needs to be for participants to reliably detect it. And participants do this on a color calibrated iPad, and they do it in this blackout tent, and that's across all 3 of our environments, so both sites in Ecuador and in the UK. So what are we expecting to see from this data? Well, if we imagine we're just talking about 2 different environments, it might be that one of our environments shows this classic elongation along blue-yellow, having a greater amount of variation along the negative diagonal than in the opposite direction. And we might have a second environment which is equal in its variation of saturation across all hues. So on that cumulative frequency plot, all the lines would be doing exactly the same thing. Whereas in this one, the blues and yellow lines would be much shallower. And if we're using efficient coding to encode our environment, then we would expect that when we look at the behavioral data from the color discrimination task, that participants who are living in this environment show discrimination thresholds which line up pattern-wise with the variability that we find in their home environment. Whereas participants who live in environment 2, where they have this different pattern, you would expect to line up with their discrimination thresholds would line up with the shape of variability found in their environment. There is an alternative option, which is that the two participants groups might share some common evolutionary history, which means that which they are aligned to, and that they don't align within their lifespan to the environments that they're currently living in. So they're not efficiently encoding their current environment, but some past evolutionary environment. So there are 2 things that we're looking for. The first is that is whether or not there's any alignment between environment and perception, so the accumulation of saturation and the saturation thresholds. And then the second thing we're looking for is whether or not the participant's own environment is the best predictor of their own perception, or whether or not an alternative environment is a better predictor. So here's the distribution of scene statistics for the 3 environments that we have. So in green, you've got the Esmeraldas, and in blue, you've got Quito, and in gray, you've got Brighton. And you can see that all 3 environments are all elongated along this similar direction, along the negative diagonal, showing a greater amount of variation along that direction than in the reflection. You can see that the Esmeraldas has this, appears to have a tilt in its orientation, which is consistent with what Mike Webster's found when he's looked at arid versus lush environments. Lush environments tend to be tilted in this way. And here's the behavioral data of the populations living in these 3 environments. So again, Esmeraldas is in green, Quito is in blue, and Brighton is in gray. And you can see that all 3 groups show this elongation along the negative diagonal. It's likely that the Brighton participants have much lower thresholds just due to familiarity with doing psychophysical tasks, rather than some other environmental difference. The important thing to look at is the pattern of discrimination. and how similar that is across the three groups. So when we compare these side by side, we can see that both the environmental data and the perceptual data both show this elongation along the negative diagonal of cowboys and chromaticity space. So the next question is, how well does each environment predict our individual participants' perception? So to look at that, we take the environment statistics and we correlate them with each of the individual participants' thresholds. And from that, we get figures like this, which show you the distribution of R values. So the correlation between these two. So a high number means high correlation, higher correlation. And if you found that an environment was particularly good at describing their thresholds, then you might expect it at this end of these figures, for example. So we do that with the participant's own environment, and then we also compare it to the distributions that you find when you correlate the two other environment statistics with each individual participant's thresholds in that population. And this is what all of those figures look like. So along the diagonal along here, you have participants' thresholds correlated with their own environment. So Esmeraldas correlated with their Esmeraldas environment, Quito thresholds correlated with Quito environment, and Brighton thresholds correlated with Brighton environment. And then the other figures show you what happens when you correlate those thresholds with the alternative environments for those participants. And the numbers which are atop each figure show you the average R value for this particular distribution. And you can see that there isn't one that stands out as being particularly different from the other distributions. And we could test that statistically by turning these R values into Fisher's Z's and then doing a Z test. So a Z test will compare whether or not there is a statistical difference in the distribution of, say, this distribution and that one. And what we find is that none of these distributions are statistically different from each other. So that means that the participant's own environment does not appear to be particularly better or worse than any other environment at predicting the thresholds of participants living in those environments. Okay, so to come back to these 2 points we were interested in, do we see any alignment between the environment and perception? And yes, we do. Both our environmental statistics and our perception data show this elongation along the blue-yellow direction from the cowboys chromaticity diagram. But the next question of whether or not participants' own environments are a better predictor of their perception than another environment, the answer appears to be no. So participants don't appear to be using efficient coding within their lifetime to tune into their particular unique chromatic distribution that they have in their environment. Their environment is no better at predicting their own thresholds than it is another environment. But we do note that there are obviously limited environmental differences between our 3 environments. So though they feel qualitatively very different from each other, they appear quantitatively to be actually more similar than you might expect. So what's next? Well, as I just said, it might be the difference between most natural environments are actually very subtle. There might not be a very big difference between them, but it'd be worth looking at a wider range of environments and trying to find extreme natural environments to see if there's a big difference that is then reflected in perceptual discrimination. Another alternative would be to make use of the artificial environments which exist. So for example, people who live on submarines for 6 months, for example, have very particular chromatic just makeup to their world that they might tune into. Another thing to remember is that that the environment might impact other aspects of colour perception. It might, within your lifetime, it might not be colour discrimination, but it might be something like preference, for example. So John Moore in our lab is currently working on a project which is comparing the way that natural scene statistics vary within an environment and how that relates to people's ratings of chromatic stimuli. And finally, it might be that just there are other environmental statistics which can better explain discrimination in particular. So it might be that the differences we find between groups in discrimination are just not captured by a biometric that tries to measure the overall distribution of chromatic variation, but by some specific measure. Okay, thank you very much for listening. We're interested to hear any questions. I just want to say thank you quickly to my collaborators and to our funders. Thank you.